It's my honor to present our work today on development of HRV-based biomarkers for predicting blood glucose levels. Around the world, diabetes has become a major health concern, and it is a chronic condition that can cause serious complications. And currently, about 500 million people are already on the edge pre-diabetes. The good news is pre-diabetes has can be reversed with lifestyle change. Therefore, regular glucose monitoring is important to measure your health. However, conventional blood glucose monitoring is invasive and often painful. That is why non-invasive blood glucose monitoring has become a key issue in healthcare technology. ECG is a non-invasive biosignal that can be easily measured using wearable devices such as smartwatches, watches, or even earphones. These advances make it possible to monitor cardiac activity anytime and anywhere. From ECG signals, we can extract heart rate variability, or HRV, derived from ECG signals, serve as a non-invasive signal. Non-invasive biomarker reflects autonomic nervous system activity and cardiovascular health. Recent studies have shown that HRV can also reveal abnormal glucose regulation in diabetic patients. However, most existing research has focused on diabetic patients, and few have explored nonlinear HRV, which can capture more complex physiological dynamics. In this study, we aim to identify HRV-based biomarkers derived from resting state ECG in a non-diabetic population. We analyzed 21 HRV features, including both linear and nonlinear indices. 30 participants were recruited according to inclusion criteria. ECG and blood glucose was measured following the protocol. ECG data was for 6 minutes during alternating eyes closed and eyes open tests. Blood glucose levels was measured and averaged from 3 commercial glucose meters. ECG data was pre-processed with a bandpass filter and downsampled. After detecting alt peaks and removing outliers, we can extract 21 HRV features using covering time and frequency and nonlinear domains. To examine the relationships, we use Pierce correlation between HRV and blood glucose. So what we found in this result is that we identified 4 HRV shows significant correlations. As shown in this figure, HF showed significant positive correlations with blood glucose levels, while the nonlinear features alpha-2, PIP, and ILS show significant negative correlations. These findings suggest that HRV reflects autonomic regulation related to glucose control, especially as HF showed positive correlations with consistent with previous study. In addition, nonlinear indices showed negative correlations, suggesting that reduced long-term complexity of cardiovascular dynamics may indicate impaired autonomic function. Overall, both linear and nonlinear features HRV may serve as potential non-invasive biomarkers for early detection of glucose dysregulation in non-diabetes. In the future, these biomarkers are expected to contribute to the development of wearable tools for non-invasive glucose monitoring. Thanks for listening.